Okay, you guys are there. Sorry about being late. So I'll shut that down. All right, so what we're going to do today is finish chapter 6 and then maybe have a little bit of review. Let me remind you there's a practice quiz number 2 due June 17th. Any questions? All right, can somebody confirm, was this the last slide we talked about?
I think it was the one before.
So we didn't talk about this last slide?
No, not yet. Yeah, we stopped here.
All right, so enrichment media. Uh, as I was saying last time, it's a media we use to enrich for specific organisms. And I think I was talking about that what you can do is you can line up several of them. So if you have one tube here, you put your culture into that, and then you let it grow for a while and it enriches, and then you take it out of that culture and put it into the next tube, which will be the same enrichment media, and that'll further enrich, and you can keep doing that a couple of times. And then you can increase the concentration of the bacteria you're looking for several times and get it up to a number where now when you grow it up, you will have a chance of getting it. So that can be particularly good when you're looking for a bacteria that's few in numbers, such as a bacteria from the soil or a bacteria from a patient's gastrointestinal tract when you're looking for one causing disease and there's many other bacteria that you have from the patient. And I gave the example like when you're looking for a phenol degrading bacteria from the soil, you just put it in a tube where the only, the only bacteria is, the only carbon source is phenol. And so that will greatly enrich for phenol degrading bacteria. And then you do that a couple of times and now you streak it out. You can find the phenol degrading bacteria. All right, any questions about that? So this slide is showing you different types of culture media. We mentioned chemically defined media. That would be where we know all the chemicals in the media, meaning that we add all the chemicals and everything we add, we know what it is. So that they call a chemically defined media. And that means that we know what all the trace elements are too, so we're not just adding tap water, meaning we're adding distilled water or deionized water, and then we're adding the trace elements. A complex media is one where we don't know what all the molecules are, and it may have ground up beef extracts or ground up cow or maybe ground up soybean. And so we don't know what all the molecules are, and we call it complex. And it supplies a majority of the nutrients for microorganisms, and a lot of things can grow in them. And that will be what most of your general purpose media will be that you use in microbiology, a complex media. And that will grow a lot of the chemoheterotrophic organisms like E. coli that we grow. And then it's just a cheap media too, so that's why we use complex medias. A reducing media is a special media we use to grow obligate anaerobes. A selective media is one where the chemicals in it select for certain organisms. And so certain things can grow in that media, but other things cannot grow in it. A differential media is one where the components of the media allow us to differentiate between two different microorganisms that grow on the media, and it'll just make the two species look differently. It could be that there's something in it that makes them have a different color. With blood agar, for example, it's the blood in the media itself, and it allows you to differentiate between the different hemolytic capabilities of the microorganisms growing on the blood agar. An enrichment media is a special type of a selective media, and it's designed to increase in numbers certain microorganisms so that they will be more easily found. All right, any question about any of that? When you're growing up microorganisms, it's important to work with a pure culture. That's because when you're running a test and the results are positive, if you have two or more species that you're working with, it's really difficult to determine which species was positive. So when you're working in microbiology, you have to have a pure culture, and that way you know what the test result tells you, and that is the species itself that you're studying is the one that gave those results. Any questions about that? All right. How you get a pure culture is generally you streak out the cultures, like if it's a mixed culture, and you streak it out on a plate and you get isolated colonies. And if a cell is streaked out on a plate, and then they're streaked out far enough away, you'll get isolated colonies. And that itself is not a pure culture. It might be a pure culture, but there can be a mixture, and so that one colony may have been formed by more than one cell. So what you do is you take an isolated colony from that plate and you streak it out a second time to get isolated colonies on a second plate. And then when you obtain isolated colonies on the second plate, it's really high chance that the isolated colony is from a pure culture. And so that's why we streak out cultures two times on a plate. And the second time you get isolated colony, it doesn't guarantee that it's a pure culture, but it's a really high chance that it's a pure culture. And that is that if you streak it out twice, let me blow that up, on a plate, the second time you grab from a isolated colony like that colony there, it's a really high chance that from that colony that went through streaking for isolated colonies twice, it's a really high chance that that will be a pure culture. And that's how we obtain pure cultures in microbiology. You just streak it out 2 times on a plate from colonies that are isolated, meaning that they're not growing together. A colony is formed by cells that are streaked out on the agar, and you can't tell if the colony was formed from one or more cells. So when we look at these colonies, we generally say that they were started by a colony forming unit, and we can't tell really if it was formed by one or more cells. I was going to hopefully look at some and show you that one colony was bigger and that might have been formed by 2 or more cells. We can say that this colony right there was obviously formed by 2 cells because we can see that when they were smaller they were 2 isolated colonies, but now they've grown together. So we say that colonies are formed by a colony forming unit, meaning that they were formed by at least 1 cell, but they might have been 2 cells stuck together and maybe even more than 2 cells. But a colony is formed by a colony forming unit. To obtain single cells, like I said, it's important to get single isolated colonies, and you do that 2 times, streaking for colony isolation. And when you do that 2 times, there's a really good chance that you'll have a pure colony, pure culture. All right, any questions about that? Streaking for colony isolation. As you notice on this slide here, when we do streak for isolation, you can see that there's at least 2 cell types, 2 different colonies, a yellow colony and then the red colonies. So that this was a mixed culture when it was streaked out, and when we isolate the first time, we think this colony would be pure because it's only showing red colonies. But to be absolutely certain, you take this colony and you streak it out a second time. And then when you isolate it a second time, then we say it's a really good chance that it's a pure culture. Any questions about that? Because you're not really going to be doing it in the lab. I think you will be doing it maybe in a virtual lab. So when we're talking about growth of microorganisms, we mean an increase in the cell number. There are 3 ways that bacteria cells can grow. They can grow by binary fission, and that's how most of them will grow, by binary fission, and that's where the cell grows and then divides literally into 2. And I'll show you a picture of that in a minute. There's also growth where some microorganisms can grow by budding. There are a few bacterial species that can grow by budding. It's the way yeast tend to grow, and that is the parent cell puts an outgrowth, which we call a bud, and that bud enlarges, and then the parent cell divides its DNA, and one portion of the DNA moves into the bud, and then the bud grows larger and then larger, and finally the bud will be about the size as the parent cell, about the same size, and then it separates from the parent cell. So one cell once again divides into 2, but instead of doing it by binary fission, it divides by budding. And I know that's not terribly clear, and we'll show a picture of that. There are a few bacteria that grow by uh, uh, filaments, and I was going to try and say reproductive spores. I was going to try and show you a picture of that where I will draw it. Oops, that's the wrong one. What we have is a parent cell, and it's usually a longer cell like a bud or a filament, and then it grows reproductive spores up at the top, and there might be several of them, at least 4, maybe 5, 6. I'm not drawing them all the same size, my apologies there. It's hard to draw here. Anyways, at one side of the cell, usually at the top, there are reproductive spores, and then when a stiff wind blows or an animal comes along, it grabs these reproductive spores and then takes it elsewhere, and then when they fall in a favorable environment, they can start growing, and the cells will probably divide in the new environment, maybe not by reproductive spores, but by binary fission. Anyways, there are a few bacteria that can grow by reproductive spores. Any question about that? And they usually grow at the tips of the filament, and that makes them easier to scatter and move elsewhere. So in binary fission, one cell, it may elongate a little bit, and then it replicates its DNA, and the DNA moves towards opposite sides of the cells, and then the cell literally starts dividing down the center. And what it'll do is it'll grow a cell wall right in the middle of the cell, and it'll have a cell membrane on it, and that divides the cell into 2. And then 2 cells come from 1 cell by binary fission. Any question about that? Over on the right, you can actually see an electron microscopic image of the cell dividing by binary fission, meaning there's the DNA. It's separated into 2 and moved to opposite sides of the cell, and the cell is literally growing a cell wall right here in the middle of the cell. Here's a picture of budding. This is yeast, not bacteria, and you can see the outcropping of the cell. And if we click on this link, you'll get to see budding happening in the little video. Let me see what's wrong here. Maybe I've got it too big. Yep. So let's try that again. So if you look at this cell here, you can see that it's starting to form a bud right there. And there the bud is puncturing out. And it looks like the DNA is starting to replicate. We can actually see that. And now the DNA is starting to move into the bud. And the bud is getting larger and larger. And about now it's going to break off from its parent cell. It's not quite the same size as the parent cell, but it's close to the size of the parent cell. And then as we go on, the parent cell is starting to put a bud over here, and this cell is starting to put a bud over there. Any questions about that? Anyways, that's budding. we talk about cells growing, we say their generation time is the time required for one cell to divide into two, or it's the same thing for a population of cells, for the time for that population of cells to double in number. Okay? So for species, it doesn't matter if that's the time it takes for one cell to divide into two or the population number to double. This varies considerably depending on the species. Some bacteria can grow as fast as 20 minutes, like E. coli, when it's growing its fastest, can grow at taking only 20 minutes for the one cell to divide into two. Most bacteria, at least the ones we grow in the lab, they grow between 1 and 3 hours. So it'll take them between 1 and 3 hours to double. Mycobacterium leprae, the bacteria that causes leprosy, can take up to 10 days for one cell to divide into two cells. And it's the slowest growing one that we know of, or at least knew of 5 or 10 years ago. This bacteria takes a long time to divide, and that's because all of the mycobacterium take a long time to divide because they put mycolic acid in their cell wall, and it takes the cell a long time to make that mycolic acid. Now, when we say the generation time is 20 minutes, it's implied that it takes 20 minutes for a cell to divide into two, or 20 minutes per generation. So that per generation is implied. It's not a time per hour, it's 20 minutes per generation. And students, for some reason, are a little bit confused when we talk about generation time. Like I said, remember when you hear that it takes 20 minutes, we mean it takes 20 minutes per generation for the cells to divide into two, or for the population to double. Any question about that? When we talk about bacterial growth, microbiologists like to use a semi-logarithmic plot, because if they use an arithmetic plot, then the time is fine, the number of generations on the y-axis. But if we use an arithmetic plot, we get this exponential curve. And all the points tend to be together, like this graph is showing you. Let me blow that up a little bit. Or actually, we can start the slideshow. You can see that one cell divides into two, two cells into four, four cells into eight, eight cells into 16, meaning most of the plots on this graph are right here. They're all stuck together. And the next graph, after 524,288, the next graph will be someplace probably on the ceiling of my room. That the points really spread out, meaning all the points are here, and then up here, after this point, they really spread out. If we use a semi-logarithmic plot where the x-axis is still arithmetic, meaning the number of generations are on this scale, and then put with the log number of cells on this scale, it spreads out the points. So there will be 1 point here, and then 1 point here, and 1 point here, and then 1 point here, and then the next point is about up right here. So it spreads out the points, and then they're more uniform on the graph. And then the other reason for using the semi-log plot is that this equation is much easier. Y equals mx plus b is much easier than this mathematical equation to work with. And so microbiologists like to use a semi-logarithmic plot where the y number of bacteria is plotted logarithmically and then the x is plotted arithmetically. Any questions about that? When we look at the bacteria growing in a population, and this will be true for most populations when they're growing in a limited environment, initially there will be a lag phase. The lag phase is because the bacteria, they come from like a solid media and then you put them into a liquid media. They're not ready to grow in the new environment. And what they're doing in the lag phase is they're not growing. They're getting the enzymes they need to grow in the new environment. And once they have those enzymes ready for growing in the new environment, the population then starts growing. So that's what the lag phase is. They're making the enzymes they need to grow in the new environment, and then they start growing in the log phase or the exponential growth phase, and they grow exponentially because there's initially lots of nutrients around, there's few cells, there's lots of space, and they grow exponentially. And then they start slowing right in this part of the phase, and that's because there's fewer nutrients around, there's less space, there's more waste products around also. Initially, there were no waste products around. Later on, there were a few waste products around. Now there's lots of waste products. And so the unlucky cells, they're not growing, and the population growth rate starts to decrease. And then we enter the stationary phase, where the population numbers become stationary. Some cells are growing, but it's equal to the number of cells that are dying. And that's why the population becomes stationary. Any question about that? The lucky cells, they have some nutrients around them, not very much waste product. They have some space. So they can grow. The unlucky cells, they've got only a few nutrients or maybe no nutrients around them. They've got lots of waste products around, not much space, and so they're dying. And the population is stationary because the number of cells dying equals the number of cells growing. And then we enter the death phase or the decline phase, where there's more cells dying than are cells that are growing. Okay? And then late in the death phase, there's no cells that are growing, only cells that are dying. Any question about that? Eventually, you'll have all of the cells die in a closed environment. Like you had a single closed test tube. Eventually, all the nutrients will be used up, and there would be lots of waste products around, and eventually all the cells would die. This could take a different length of time depending on what the cells are and what species they are. If you have a species that produces endospores, this could take centuries, okay? Most bacteria that don't produce endospores, all the cells would die after a few weeks, maybe as soon as 2 weeks, okay? Like I said, it'll depend on the species and how hardy the species is before all the cells die. All right, any question about any of that? Oops, trying to get that a little smaller. When we're talking about cells growing, there are different ways to grow the cells. There are different methods to measure the growth of microbial populations. And this slide is showing you that there can be direct methods where you're directly counting cells. And then there's indirect methods where you're getting a basis on the number of cells, but you're not directly counting them. Is that clear? The direct methods and the indirect methods. Okay, so the direct methods, you can do a plate count, often called a viable plate count. We'll talk about that more. Filtration, where you filter the cells. So that would usually be a liquid media, and you send it through a filter. And I don't know, like you can filter a gallon of cells, and then you're left with just the cells stuck on the filter, and then you put the filter on the media, and you allow it to grow, and then you count it. There's the most probable number, and that is the one way I've actually never done a count. All the other ways I've done a count. And anyways, the most probable number is a direct method of counting cells. There's the direct microscopic count, where you look at the cells under the microscope. You can measure the turbidity of a solution, and that will be an indirect method of the number of cells. You can look at the metabolic activity, and if there's more activity, metabolic activity, then you say there's more cells. Or you could centrifuge the cells out and let them dry and get the dry weight, and that will be a measure of the number of cells. So let's talk about each of those more. The direct plate count, we'll also call the viable plate count, is a very common method used for obtaining the number of viable cells in a solution. And you plate the count cells on a media, and then grow them up, and then look for the number of colonies that grow, count the colony forming units, and then you say that's how many cells were viable in the solution that you plated onto this plate. This is the only method that counts viable cells, because only a viable cell will grow and form a colony. If you have a dead cell there, it will not grow to form a colony. So this method counts viable cells. In a plate count, like I said, it's a common method to do, but you just can't take a solution and put it onto a plate. You have to make a serial dilution, making several different dilutions, and then inoculate each dilution onto the plate. Spread that solution out on the plate, and then grow it up, and then base your colony counts on that dilution that you plated on the plate. The dilution that you put on the plate. Okay? Any questions about that? So how you make a serial dilution is illustrated in this plate. This slide, you have 9 mils of broth in each of these two, and from your initial solution you put 1 mil in here, and now you have a dilution of 1 to 10. You mix that tube up, and then you take 1 mil out of that and put it into 9 mils, and now we have a dilution, another 1 to 10, so this will be a total dilution of 1 to 100, meaning it's a dilution of 1 to 10 here and a dilution of 1 to 10 there, so it's a dilution of 1 to 100. You mix that up, take 1 mil out of it, put it into this 9 mils, and now you have another 1 to 10 dilution, so this is a dilution of 1 to 1,000. Continue that 1 to 10,000, 1 to 100,000. You take 1 mil now from each of these, possibly also from the original inoculum if you don't think there's too many cells in it, plate it onto a petri dish, and I'm only going to talk about plating 1 mil onto the plate. You could plate 1/10 of a mil onto the plate and then spread it out. This one you're just using gravity to spread it out, but you could spread it out also with spreader, and then you incubate it and then allow the plates to grow. Any question about that? So they usually grow for at least 24 hours. And then we have our different dilutions, and you note that only some of them have countable colonies, where you can count this amount and you can count that amount. This one, there's too many cells to count. Let's see if we have a number there. It doesn't look like there's a number there. Oops, too big, now too small. Boy, that's really going large. But anyways, in this plate, and this plate, and this plate, the number of cells we put on the plate were too many to count, so you just put it 2M, 2C, too many to count. And this one, you can count it up, and let's see what that is. It looks like it might be around 30 cells, maybe 33, I don't remember what it is. And that one is easy to count 4, but what you want to do is count the plate where there's between 30 cells on the plate and maybe 300, more than 300 or maybe more than 250. There's too many to count, and so the count is thrown off, like we don't know how many colonies are in this blob right here. The point is we want to count where we have a good count, and on this plate here, which only has 4 cells, well, if you have plus or minus 1 colony, that's plus or minus 25%. And if it's got 30 or more, that's plus or minus 1/30, which is something like plus or minus 3.3%. So the mean is a much better mean on this plate if we take the count from this plate than we do from this plate, because this one, like I said, plus or minus 1 is just too big of a variable. So we do our count when we can get a plate where we can count between 30 and about 300. And that way it's just a better count. Any questions about that? How we get our viable plate counts? All right. If you had a very low number of cells in a solution, you can send the solution through a filter, as shown here, and then put that filter on an agar plate, and then allow the cells to grow. And as you can see on this filter here, you can count those. I don't know, that's an awful lot. 100, maybe even 200. But the point is, if we only got 100 or 200 cells per gallon, for example, we would have to send it through a filtration and then make our count of cells. This one, when we look at the filter here, because that's microscopic, if you were to grow those up, all of these colonies would grow as one, so this filtration would not be countable because there's too many cells that are plated on the filter here to get them to grow up into colonies. I suppose on this one you'd have to look at it under the microscope to count the number of cells per mil. Any question about how you obtain the colony count from a filtration? Like I said, you use a filtration when the bacteria numbers are really dilute in your solution. A direct microscopic count is where you take a volume of the liquid, and it's a measured volume, and then you look under the microscope to get a direct count on the number of cells. That is not good for motile bacteria because they will be moving, and it'll be very difficult to count if they're mobile. And you cannot distinguish between a live, viable cell and a dead cell because they'll look similar under the microscope. You need a high cell concentration to do this unless you centrifuge the cells, because if the cells are too dilute in the volume and then you put it under the microscope, you might not see anything under the microscope. But the advantage is there's no incubation time that's required for colonies to grow up. When you're doing a direct microscopic count, you have a special slide, and it takes a special volume directly under the cover slip. And usually what it's set up is that this square here, whatever number of cells you see under this square, you then multiply this by 1 million, because this volume here under this square right there is 1/1,000,000th of a mil. So what your count would be whatever this count is, maybe that's about 15 or 20, and then you multiply by 1,000,000, and then you say that there are 15 to 20 cells per milliliter of the solution that you counted. Any question about that? This is a specific slide that takes a specific volume of liquid. And the volume under this square would be 1/1,000,000th of a milliliter, or about 1 microliter. Any questions about that? Like I said, this is advantageous because you can do it right away, although there are some disadvantages. Obviously, some poor schmuck has to do the count, and you can't tell is that a viable cell or a dead cell because they look the same under the microscope. And I've done many counts like that in my day. Anyways, those are the direct methods of counting cells that I'm going to talk about. There's also indirect methods for counting of cells, so let's talk about the indirect methods you can use for counting cells.
Are we going to do the second meeting today, or are we just going like to 3:45?
Is it time to stop?
It's 2:20.
Okay, yeah, you guys have to tell me. I'm not looking at the my computer here, so I didn't know it was 2:20. All right, let's go ahead and stop this, and we'll come back. Well, actually, we're almost done here. I'm on my last slide, so second to the last slide, so let me go ahead and finish here. And then we won't come back for the second meeting. Any question about that? All right, so turbidity is where you measure the volume, and what you're looking at is you're looking at the light coming through the solution, and when there's a cell, that will block the light. So if you have cells in this tube here, less light is going to get through, and that will be measured compared to the solution initially where there's a full amount of light getting through. And so you can check this against a known standard, meaning you got a chart, and the amount of light getting through will be dependent on the chart for how many cells are in the milliliter of the solution. And so by turbidity, the amount of absorbance of the light, we can actually get an estimate for the cell number. Any question about that? The more cells, the less light will get through. The fewer cells, more light will get through. And then you look on a chart, and that tells you how many cells per mil you have. And obviously you have to make this chart, but once you have it made, then you can always use that chart for getting your estimate for the number of cells per mil. And this is a very easy way to get an estimate of the number of cells. You just get a tube of it, put it in, and then do your reading of the on the spectral photometer and look on the chart, and it tells you how many cells per mil you have. So that's a very easy way of getting the number of cells that I've used. The next slide shows you two ways where you can get an estimate of the number of cells. An indirect method. You can measure the metabolic activity of the cells. Like you might measure the amount of CO2 coming off of the cells. You measure how much CO2 is coming off, and the more CO2 there is, the more cells there are. Okay, and then you look on a chart, and then it gives you an estimate for the number of cells there are from the metabolic activity, like the amount of CO2 that is made. Any question about metabolic activity? And then dry weight is something similar, except you centrifuge down the cells, allow them to dry, and then at your convenience you weigh the dry weight. And the more cells there are, the more weight there are. The more cells there are, the higher the dry weight will be. And then you look at a chart and it gives you the estimate of the number of cells. Now, this method in particular, you can't tell the weight of dead cells versus the weight of viable cells. So you can't tell on this one whether the cell is living or dead. On the metabolic activity, if it's a dead cell, it won't produce the metabolic activity, it won't produce the CO2. So actually this one is measuring viable cells. All right, any question about the two ways for getting an estimate from this slide, estimating the bacterial numbers by indirect methods? Okay. If not, we're not going to meet at 2:30 because I've finished this lesson now. All right? Any questions? If not, I will see you tomorrow. And my apologies for starting late today. If you didn't get this, I will put this up and then you can see it on my YouTube site. All right, I will see you tomorrow.